So, in general, you know, when you bring things into the hood, you find some way of sterilizing them first. And that's why I was spraying the bottles down with ethanol before I was putting them in the hood.
So are the markers sterile too?
They've been cleaned. I don't know that they're perfectly sterile, but they're clean.
Okay. Which one are you doing right now?
So these are the cells that they're not so confluent that they need to be split. They're very dispersed on the surface. You know, we don't need to clean them up. So all I'm going to do here is just replace the media so that they have a fresh media to grow in.
These don't need to be split. Is that what you said?
They don't. So these little glass pipettes, of course, are sterile. And what I do is I'm just basically aspirating off the solution that the media that they're bathing in, and then the cells, because they're adherent, are left behind. So they're sitting on the surface. The reason why I was making different stacks is I was trying to keep myself clear on who needed to be split and who didn't. So I do little things that you may or may not notice or care that much for, but little things like to keep the cells clean. So for example, like you see when I lift the top off,
yes.
I don't ever wave my hand over top of the top of the open container, and I don't touch the inside of the top. So I spin it like that, but I don't touch the inside of it, and I put it like that so it's facing up. And then so I keep my hands away from the top, from over top, so nothing falls into the cells. And I aspirate off. So there's a little bit of kind of technique to it. It's not too hard. You learn it in about a week, and then you don't even think about it anymore. It's like riding a bike, you know? These are called pipettes. These are just the things we use to transfer solutions from one thing to another. Thank you, man. What's up?
It's really quiet around here today.
It is. What's going on? I kept thinking I'm like missing a meeting or something.
It was quiet on the TV on my way in too. And I was like, I mean, I know you take the commuter rail, but was it different there?
It was quiet on the TV for us too. Yeah. I feel like we missed out on some news. There's been some nuclear holocaust somewhere, and
Everyone's mourning.
yeah, we're sitting here doing our cell culture.
What do they call it? The rapture.
The rapture happened. we think we got saved, but actually we may be the biggest victims of all. So Brian, this is Rachel, and there are two high school students,
Ah, okay.
and they're here with my sister.
So
And
where's your sister live?
I didn't say Rachel and Megan, and they're two high school students. My sister lives in Kentucky,
Okay.
and they're here with her, and they're seeing how science is done, at least in as much as I'm doing science.
Megan, you guys are in European live in Kentucky?
Yes.
So Brian is from Buffalo, New York, originally. He did his PhD in Texas at
This is Brian.
a place called MD Anderson, which is a super famous cancer hospital. You guys may have heard of it before. Brian's kind of a big deal.
Very kind of you, so. Kris is the big deal. So which one is which?
I'm Megan.
I'm Rachel.
Sorry, we have a video blog and we're videoing this for part of our blog.
Are you really?
Yeah. So this is Brian.
Can we ask, like, what are you doing?
Sorry.
Okay.
No, Brian's actually so like of everybody in life. Brian's like the one who like if you want to learn something, you ask Brian because he's like, extremely good at like teaching things to people. And he's got all these undergrads who work for him, and every day you can hear Brian at the whiteboard in the hallway. Now and then so and so. So what would you do? And then the kids are like, I think I would. So generally, when I don't understand something, I go and talk to him. So these are the suspension cells, right? So I was telling you that they don't stick.
Right.
So what I'm going to do here is I'm just labeling two tubes. And then the way that I get the cells out is just
You might have to put
separate them out. Okay. So are you giving group meeting this week?
Next week.
What's that?
Next week.
Oh, next week. So every week, yeah, it's so much fun. You spend your week working on a talk. So every week, our lab, which is about 25 people or so, has what's called a group meeting, where one or two lab members will stand up and give a presentation on what they've been working on. So about every 3 or 4 months, it comes to be your turn to give a talk. And that's one way of not only helping everyone to know what everyone else is working on, and to provide a way for people to communicate with each other about their science, but also it's a way of, I think, keeping people's projects moving along efficiently, because, when you have to give a talk, you really make sure you have a lot to talk about.
I feel like I'm in your space. I'm sorry.
No, it's good. So that's Thursday mornings. I've been missing a lot of group meetings lately because I've been traveling. But that's something that almost every, high-powered labs have group meetings every week, It's a very ubiquitous thing.
So do you have to do this for all of them?
I do.
All right, we're ending the video here. Now this is for the enzymes.
That's right. So what we're doing is first we're getting rid of the old media that the cells were sitting in, because they've been sitting in this for about 3 days. They've depleted most of its nutrients, and we just want to get rid of it.
Okay.
Now, if the cells, like I showed you in the last batch of cells, if they haven't grown enough, there aren't enough cells in the dish, then you wouldn't worry with splitting them. You would just replenish their media and put them back in the incubator, and they're fine.
Okay.
In this case, since they've grown enough that they need to be split, we use this enzyme treatment. The first thing is we remove the media. And you can see the cells?
Yeah.
How it's cloudy?
Mm-hmm.
So that's a reflection of the fact that there are a ton of cells on the surface. So now I removed all the media. Now I'm gonna rinse the surface of the cells with PBS, which is phosphate buffered saline. It's just like salt water. It's pH 7.4 because that's what our body's like. That's the pH of like your blood and a lot of the fluids in your body. It's what the cells like the best. Our media is also pH at 7.4. 3, 4, 6. Okay. So I'm just going to add a little bit of PBS to each one of them. And what this does is it removes the old media, but what's important is that the media that the cells are cultured in has a lot of protein in it. We actually take serum from cows and mix that into the media before we add it to the cells, because that contains proteins like growth factors that they need to survive, insulin, things like that the cells need. But there's a lot of protein in that media, and so if we tried to leave the media on top of the cells when we add the enzyme trypsin, the overabundance of protein present in the media would prevent the trypsin from being able to cleave the cells off the surface, because there's so much protein just floating around that it would basically occupy all the trypsin. It would saturate it. And so the reason why we remove the media and then wash the cells is to get rid of all the protein in the dish except the protein that's sticking the cells to the surface. And in that way, when we add the trypsin, it's going to act specifically on the protein that we want it to act on, if that makes sense.
So but this doesn't take the cells away from where they are right now?
Nope, just rinsing them off.
Okay.
Yep, they're still sitting there. And they're actually really stuck. Like, they really, you know, if you took something and, like, if you took a razor blade and you tried to scrape them off, you could scrape them off. You'd probably tear them and stuff while you were doing it, because they're pretty delicate.
Yeah.
But yeah, they stick really well. If you, like, for example, if you just tried to like spray them with water, it would be hard to get them off because they really like to stick well. So you don't have to worry about them coming off.
Okay.
What you do have to worry about is adding trypsin for long enough that they actually will come off. Stupid thing. Okay. So we need 1 milliliter of trypsin for each dish. I'm using a small tip this time because I can get more precise volumes. 1, 2, 3, 4, 5, 6. You might notice that I never ever touch the tip to anything, especially to the cells. If I did that, if I accidentally was doing this right here, right, and putting this in, if I accidentally went like that, then I would not use this pipette anymore for any other cells, and I wouldn't use it to go back in the trypsin because, in theory, it could be contaminated with the cells that it touched. So there are little considerations that you're always making when you're doing these things, when you're working in the hood, just to prevent not only contamination of the cells, but cross-contamination from one batch to another. So while this is going, it takes like 5 minutes. I'm going to grab the other media.